Hello students, welcome to EPG Pathshala. I Dr. Vikas Rana from Punjabi University Patiala. Today we are going to discuss on module ratio optimization of different components for the development of stable microemulsion and its thermodynamic aspect from your paper Novel Drug Delivery System. Microemulsion consists of three main components. One is oil phase, water phase, surfactant phase. Sometimes oil phase is replaced with lipid phase also, and sometimes combination of surfactant to co-surfactant are used. Therefore, to optimize all these three components together will results in formation of a optimized microemulsion. Now it is very difficult to optimize these three or four components together. In a conventional technique, we have keeping all the three parameters constant and we can vary one if we have four components. However, if we have one component and we can vary other three components together, it will give us a large number of experimental trials to be conducted for this particular purpose. Therefore, there is a need to go for an alternative technique. This alternative technique provides us a good optimized microemulsion system in such a manner with minimum number of experiments, so that we came to know that we have optimum amount of oil phase, water phase and surfactant phase together. In some cases, if it is required to add co-surfactant, which we can easily incorporate a co-surfactant system, and there is no need to go for a conventional system whereby we can vary one part and keeping other part constant. Therefore, there is a technique which is called as optimized technique is required. So students, let's see what we are going to learn from this module. We will understand the ratio optimization technique available in the literature for the development of microemulsion and study its thermodynamic aspects. If we see the literature, there are mainly two techniques which are given in the literature. The first technique is phase diagram technique. One of the very famous technique is phase diagram technique, and this phase diagram technique is now joined with quality optimization technique, which is known as QBD. And this QBD is so helpful that we can easily get at least one optimized formulation of microemulsion. Phase diagram technique mainly involves pseudo-ternary diagram or ternary phase diagrams. In pseudo-ternary diagram, we generally use a combination of surfactant, co-surfactant and water phase also. Whereas in case of ternary phase diagram, we have combination of surfactant, water and oil phase. Therefore, these techniques will provide us a microemulsion region, and this region we can utilize and keep them into a quality by design approach. And this quality by design approach will give us a center point so that we can easily predict the optimized formulation. The main techniques which are involved in the QBD, they are factorial design, mixture design. Mixture design consists of a simplex lattice design, optimal design is there. Then we have one more very important design which is called as a central composite design and box Bancom design. All these designs are nowadays very well used for the purpose of applying quality by design so as to achieve a good optimized formulation. To understand what actually this technique, let us now discuss we have phase diagram technique. This phase diagram technique is nothing but it has taken from you can say mathematical models, where we can use equilateral triangles for the purpose of you can say our study. The diagram represents the equilibrium between the different face formed, that is when the three components of a triangle, they are act as one of the you can say three components of a microemulsion. And these diagram predict that these three components when mix in you can say different proportions, and these components will give us say a function of clarity, or a function of particle size, or a function of a temperature. They are taken into account, and then we'll achieve a single phase diagram. This phase diagram will give us microemulsion region. Phase diagram is equal to equilateral triangle, and it is also called as Gibbs triangle. This triangle constitutes three corners of a triangle, that is A, B and C, and these corners are given the components of a microemulsion. That is, if A constitute a water phase, B constitute oil phase, and C as a surfactant phase. Therefore, these three corners will combine, and the points which are located inside, they will give you different proportions of a microemulsion to be prepared. There are two types of phase diagrams. One is ternary phase diagram and another is pseudo ternary phase diagram. The main difference between ternary phase diagram and pseudo ternary phase diagram is the way you are going to take a components. For taking three components of a Gibbs triangle, that is called as ternary phase diagram. Whereas when you are taking four or more than four components for the optimization purpose, then that diagram is called as pseudo ternary phase diagram. For example, if A is equal to surfactant, B is a water phase, C is a oil phase, in this case this is called as ternary phase diagram. Whereas when we are going to take surfactant is to co-surfactant, that is called S mixture, in one corner of a Gibbs triangle, B constitute a water phase and C constitute a oil phase. In that case, total for the optimization purpose, we have four components. Therefore, it is called as a pseudo-ternary phase diagram. Instead of taking S mixture, we can also take a drug or combination of drug and S mixture in one of the corner of a Gibbs triangle. Now in this image, you can see different type of phase diagrams. This diagram is between, the diagram 1 is between capryl 90 water and tween 80 is to TCP. TCP is transquitol P. So we in this case, as a C corner of a Gibbs triangle, we have taken surfactant is to co-surfactant proportion, that is 1:1. So here is the two components. In the second phase diagram, you can see in the C part of the Gibbs triangle, the proportion is between surfactant to co-surfactant, that is tween 80 to transquitol P is 2:1. In the third Gibbs triangle, the C corner is tween 80 is to transquitol P is 3:1. In this way, we are optimizing total four components, and that is why this type of diagram is called as pseudo-ternary phase diagram. Important thing is, you can see the area, the black colored portion marked on these Gibbs triangles is indicate the microemulsion region obtained by taking all these three or four components of a phase diagram. This is another example which you will see different types of phase diagrams, and here we have A as capryl 90 water, and as well as the different proportions of surfactant to co-surfactant are taken, whereas surfactant is solutol HS15, co-surfactant is transquitol P. You can see in all these three Gibbs triangles, the inverse first case, the solutol HS15 is to transcutol P ratio is 1:1. In the next case, it is 2:1. In third corner, it is 3:1. Therefore, this type of phase diagrams, they are called as pseudo-ternary phase diagrams. And important thing is, when you increase the proportion of SHS15, that is solutol HS15, there is a increase in there is decrease in microemulsion area. Therefore, this type of diagram is called as a pseudo-ternary phase diagram. Now this example is example of ternary phase diagram, whereas we have taken each component in the three corners of a Gibbs triangle. In the first triangle, the solutol HS15, transcutol P and ethyl oleate are taken, whereas the microemulsion area is very large. In the second Gibbs triangle, you can see ethyl oleate is replaced with Jalusair 44/15. In fourth triangle, you can see the replacement with Miacin 35. And in last Gibbs triangle, you can see solutol HS15 is replaced with Labrasol. So in these four Gibbs triangles, the only three components of a microemulsion are to be optimized. Therefore, such type of diagrams, they are called as ternary phase diagram. And one important thing to note here is that the maximum microemulsion area which is obtained is with the solutol HS15, ethyl oleate and transcutol P combination. Now the question is how to locate composition of point X on phase diagram. Now we have taken arbitrarily point X at the center of the Gibbs triangle, and you can draw a line passing through X and parallel to each of the sides. Similar lines were be drawn parallel to each side of a triangle. Now on drawing a line passing through the point X and parallel to line AC, you obtained a line A double dash C double dash. Then line parallel to AB is drawn, which is labeled as A double dash B double dash, but it should pass through point X. Then again draw a line BC parallel to this BC line, that is B double dash C double dash, but passing through point X. Therefore, the proportion of each component on AB coordinate can be labeled as 40% for component A, 40% for component C and 20% for component B. This is the composition of point X. This results indicate that the corner of the triangle which marked A is towards the corner C, and the point which is at the corner C is marked towards the point A, and the center point is constitute the B component. Now the question is how to construct a phase diagram by water titration method. For this preparation of phase diagram, you can take different ratios of surfactant is to co-surfactant and prepare a vials. Then for each vial you have to add different proportion of oil is to surfactant is to co-surfactant, that is oil is to S mixture. So we take for example 90:10, 70:30, 50:50, 30:70, 10:90, 80:20, 60:40, 40:60, 20:80. And then these vials were then titrated with water under magnetic stirring to achieve homogeneous dispersion or solution. In this process, you have to check for each vial while titrating that there should be appearance, clear, turbid, or translucent or gel-like, as well as you have to look into flow properties. The end point of the titration is considered as cloudy or turbid. That is, up till that you have to titrate with water until you have achieved cloudiness or turbidity. You have to record during this process that the quantity of the aqueous phase required to make the mixture turbid should be known. In this table, you can easily see that we have prepared different self-emulsifying mixtures, that is a proportion of S mixture, which is 1:2 or taken as bridge 7 to 1 is to ethanol composition, oil phase and water phase, and these different self-emulsifying mixtures were prepared, and then the end point, that is evaluation point, is taken as self-emulsification time, appearance, particle size and drug content. So it is important to note here that if while titrating we have to take into account what is the color of the vial, that is white, bluish white, clear. So these type of appearance should be used to examine the different behavior of the S mixture or self-emulsifying mixture. Now to identify different textures which are obtained while preparing a phase diagram for the optimization of microemulsion, Winsor classification was used. Winsor classification is on basis of the type of area or the region you have obtained. We classify that Winsor 1, Winsor 2, Winsor 3 and Winsor 4 microemulsion areas. Now to further understand what is Winsor classification system, Winsor 1 is an area consisting of not only microemulsion region but also a region which is specified as oil rich area. In Winsor 2nd, we obtain microemulsion area but a small area which is water rich area and is called as a surfactant poor water phase. In Winsor 3, oil rich area, microemulsion area as well as the water rich area appears. So oil rich area constitutes surfactant poor area, whereas in which the microemulsion region is obtained is called surfactant rich area, whereas if water rich area obtained, in that case it is surfactant poor area. In Winsor 4 classification is the system, the phase where whole of the area is microemulsion area, and it is called as single phase homogeneous mixture, which is called microemulsion area, and all the components in a microemulsion area are in good proportion, which are useful for the preparation of microemulsion. In this example, you can see different oil phases were taken. In first triangle, the oil phase is ethyl oleate and turmeric oil, whereas in the second triangle you can see ethyl oleate and clove oil. So you can see there is a difference in microemulsion region when we have titrated with water and surfactant. So this image revealed that area 35 is higher, which is given by turmeric oil as compared to the clove oil. Therefore, the phase diagram provide a brief range of constraints, that is levels of oil, surfactant, co-surfactant and water mixture, that upon mixing gives a microemulsion area in a particular microemulsion. Therefore, specific single best possible optimized composition of containing oil phase, surfactant to co-surfactant and water, that gives microemulsion. Microemulsion it has to be, you can say, obtained. However, in this particular phase diagram technique, the main drawback of this particular phase diagram technique is we doesn't get a single one optimized microemulsion formulation. So for that sake, it is therefore important to shift these values of constraints, that is lower level to a higher level of these oil surfactant and co-surfactant water mixture to be put into that system which is called as quality by design approach. And this quality by design approach will give us optimized formulation after getting required using these required softwares. Thank you. QBD is called as quality by design approach. This quality by design approach is utilized when the constraints of different oil phase, surfactant, co-surfactant and water were obtained using ternary phase diagrams or pseudo ternary phase diagram. Using these constraints, we have equate on that of the evaluation parameters necessary for the good microemulsion, that is droplet size, zeta potential, drug entrapment efficiency, thermodynamic stability. All these parameters along with that of the constraints which are obtained from microemulsion region, these are equated, and the best formulation, that is optimized microemulsion formulation with lowest droplet size, highest drug entrapment efficiency and thermodynamic stability were determined using computer softwares. In this Gibbs triangle, you can see a microemulsion area, and these microemulsion area constitute constraints of utilized for the quality by design approach. And then these point of region is then studied further using quality by design methods. The question is, what are quality by design? So we have different list of quality by designs, that is factorial design, mixture design, which is composed of simplex lattice design or optimal design, and then another is central composite design or box Bentham design. These are the designs which are available in the literature and utilized for the preparation and optimization of microemulsion. Utilizing these designs, we apply a technique called graphical or numerical optimization technique, and it is joined with that of the computer softwares to provide a good optimized formulation as per our need. For applying quality by design approach, it is very important to understand the different steps which are involved in the application, in the use of these quality by design approach. The step first is the selection of independent variables. These variables act as input for the product as well as that of the process variables. And the second stage in this is selection of dependent variables. Dependent variables is reflecting the quality of microemulsion you are looking for. That is why it acts as a output product or process variables. For example, if you are looking for a very good, say, microemulsion, what you are looking for? You are looking for a lowest particle size, having a good self emulsification time. So these type of parameters are to be considered in dependent variables. So you have to select dependent variables on basis of which these parameters that microemulsion ultimately you will obtain will involve in these dependent variables. The third stage is selection of suitable experimental design, which is one of the very critical, you can say, a stage in this quality by design applications. where to use Simplex Lattice Design or where to use a factorial design. For example, factorial design is used for the selection of different factor influencing studies if you want to conduct on a particular, you can say, formulation combination, and so that we can get a very good, you can say, microemulsion. And selection of experimental domain is another one of the big criteria to map the response on the basis of the study objective is the main idea behind the experimental design. The stage fourth is selection of optimized formulation. So we select the optimized formulation using our computer softwares, and these computer softwares on basis of graphical optimization technique or using a numerical optimization technique, it is quite helpful in getting a particular one or two optimized formulations. So these obtained optimized formulations are then used for further, again you can prepare these formulations, whatever desirability levels, whatever the approaches you have obtained, what are the parameters which you have taken for the optimization formulations, you can note down the predicted values and compare it with that of the observed values. So that stage where you compare predicted value and that of the observed value is labeled as a validation of responses. So validate the responses and ultimately you will get the computer, what computer has given to you and what experimentally you have get it, that can be compared at one level, and then you can achieve one of the best, very good formulation as per your desirability. Now the question is how to select active variables and factors for the optimization of microemulsion. Using the ternary phase diagram, we have obtained different microemulsion regions and we have applied these constraints obtained using determination of microemulsion region, and then these regions were put on, they are taken as an experimental domain for quality by design, and on this basis we select active variables. Active variables are composition, concentration or level of oil phase, level of surfactant, level of co-surfactant or its mixture, whichever the case which we have taken for determining the microemulsion area. And then different experimental domain were developed depending upon the experimental design, that is a square experimental design or spherical experimental design such as central composite design are taken into account, and then accordingly we select a factors, that is dependent variables, that is particle size, drug entrapment efficiency, etc. are to be taken as the factors which are responsible, independent or dependent factors which are responsible, and these dependent and independent factors were compare each other using computer software. Now each factor has different levels. Depending upon our experimental domain, we either take two levels, that is upper limit or lower limit, or three levels, that is upper limit, middle limit or lower limit. So the variables may be either qualitative or quantitative, and these levels were labeled as -1, 0, +1, that is for lower, middle and upper respectively. Then multiple linear regression between factors with their coded variables and response variables will give in the form of an equation. This equation is y is equal to b0 plus b1 x1 plus b2 x2 and so on, where b1, b2 or b7 are the coefficient of a factors, x1, x2 and so on, whereas b0 is the constant which represents the true or theoretical response. As per these equations, it is very difficult to come out with these equations manually. Therefore, it is nowadays we have availability of computer software. So utilizing these computer software, the job is very much easier nowadays. Therefore, we have design expert software which we can utilize for getting these equations so that we can easily predict a correlation between dependent and independent variables. To further clarify the concept of application of quality by design, we have taken an example of Simplex Lattice Design for the formulation optimization of microemulsion. Now you can see there are independent and dependent variables. Independent variables are put in the form of a design which is available in the literature. 0 constitute a middle phase, 0.667 or 0.333 are the intermediate phase, and 1 is that of the highest level. In case of dependent variables, we can take dependent variables as y1, y2 and so on, and these independent variables or dependent variables, they are then equated. The dependent variables are, for example, particle size, zeta potential, thermodynamic stability, self-emulsification time of a microemulsion. These dependent variables are selected as per our need, that is, whichever dependent variables which we require or whichever properties which we are looking for in a good optimized microemulsion. And then the multiple linear regression analysis using computer software is then employed to achieve correlation between dependent and independent variables. Now this diagram is a triangle that is called equilateral triangle and is showing a different points marked on uh, as per Simplex Lattice Design, and it is called as experimental domain. You can easily see where the points 0.667 or 0.333 located on x1, x2 and x3 coordinates. In another example where extreme vertices design is used to optimize the components ratio of SNEDDS, that is self nano emulsifying drug delivery system, where surfactant component, co-surfactant and oil component are optimized for the preparation of microemulsion, and the requirements are in the form of dependent variables, that is solubility and droplet. And the aim is we are looking for a microemulsion which can give us highest solubility of a microemulsion with the smallest droplet size. In another example, Simplex Lattice Design for the preparation of microemulsion is utilized, where oil phase in microliters, surfactant in microliters and co-surfactant in microliter were taken as independent variables, and these points are at the axial scale, third edge, vertex, center, and these points are taken of a equilateral triangle, and the response is taken as droplet size, PDI. PDI is polydispersity index, and self emulsification time. And then after multiple linear regression, we have obtained a statistical analysis in the form of a model, that is cubic model or a quadratic model are obtained. In this case we have obtained cubic model, and then R square values, that is how much is correlatable our this correlation between dependent and independent variables are. And then using a numerical optimization technique we obtain a desirability. Desirability is, that is what we really require, that is how much is the droplet size we require, that is minimum. We require PDI minimum, and emulsification time should be in the range of our experimental domain, so that we achieve a good desirability. On this basis we have obtained a desirability near to 1. So our aim is to achieve a desirability near to 1. Therefore, we obtained different formulations using a numerical optimization techniques, and these formulations L1, L2 and L3 were then further evaluated, prepared and then evaluated for droplet size, PDI and emulsification time, such that the correlation between the predicted values by the software and the one which I have observed values, that has to be correlated with each other so as to obtain a good optimized formulations. Now this is one of the researcher has utilized central composite design for the preparation of microemulsion, where factor 1 is a lipid phase and factor 2 is surfactant phase. They are then coded and then utilized for the preparation of self-emulsifying drug delivery systems as per central composite design. One of the important aspect of microemulsion is its thermodynamic stability. To understand what is the process of formation of oil droplet, that is, look into thermodynamic stability. If we take a bulk oil phase and that bulk oil phase is converted into oil droplets, there is a change in entropy, and also there is a increase in surface area, which means increase in area means negative surface area change, that is delta A, and this will affect on the equation which is called as a free energy of formation, that is delta GF, which is equal to the surface tension into delta A minus entropy change, that is T delta S, where T is temperature and delta S is entropy. So this equation is one of the very good equation that can be utilized in understanding the thermodynamic stability of a microemulsion. Now, to further utilize this equation, you can see the two components of this equation are treated as two corners of a stick, and then if there is an increase in delta A, that is negative delta A, and reduction in surface tension at the same times, that leads to increase in entropy change. This will increase in stability, and the delta G value leads to negative, and negative delta G means stable microemulsion. In another case, if gamma delta A, that is surface tension into delta A, is increased and entropy change is reduced, in that case we obtain delta GF from that equation as a positive value, which means the macroemulsion formation and which is kinetically stable. This is the diagrammatic representation of free energy as a function of droplet size. You can see when there is a negative delta GF, there is a stable droplet size and microemulsion is formation takes place. Whereas if there is a positive delta GF, there is a macroemulsion formation, or in C case there is no emulsion formation if there is a increase in the diameter of the droplets. Therefore, there is a limit that at one particular point there is formation of macroemulsion, and if the droplet size increased, there is no emulsion formation because of the positive delta GF. So students, let's summarize what we have learned from this module. The different components of a microemulsion are optimized by employing phase diagram technique, following by quality by design approach. The ternary phase diagram is prepared for the optimization of at least three components, whereas pseudo ternary phase diagram is for 4 components or more than that for the optimization. And in end, QBD approach is for achieving best possible optimized formulation with all required properties.